In this video, we're going to talk about how the cell regulates the cell cycle. As we've seen, there are a lot of events that have to occur correctly for a cell to divide. It has to replicate its DNA properly. It has to do all the steps of mitosis properly. It's a complicated process, and regulating it is just another complicated layer to this cell cycle. So if you remember, we talked about the fact that cells, if they're not dividing, are hanging out in G0. Okay, but what we haven't talked about yet is that the cells will then be called out of G0, and there are going to be certain checkpoints along the way that will check and make sure that everything is going okay. So in this video, we're going to learn more about how the cells know when to divide and how they know that they're doing the steps of the cell cycle correctly. So we're going to start by going through a series of questions. So our first question is, how does the cell know when it's time to exit G0 and continue with the cell cycle? So if the cell doesn't receive a signal, okay, it's going to grow in G1 and it's going to basically just do its normal everyday activities. If it doesn't need to, it's not going to progress on to this through the cell cycle to the S phase, and it'll just hang out in G0. So there has to be a signal that gets received by the cell to tell it it's time to go ahead and divide. So that signal is an external signal. And these external signals are one of two types: growth factors and growth hormones. Growth factors are often used in repair. So let's look at an example of this. So let's say you grabbed a piece of paper and you got a paper cut. Ouch. So your producer cell in this case is going to be a damaged skin cell. So it may have gotten cut or damaged by the paper, and as a result, it's going to need to communicate with other cells within the skin to coordinate so that new cells can be made to fill in the gap from where the paper cut is, because it's an emergency. A paper cut may not seem like a big deal, but that is an open wound, and germs can get in and can cause serious infection. So your body needs to quickly close it up. So that damaged skin cell is going to release a signal molecule. In this case, it's going to be GF, a growth factor. Okay, and that growth factor will then tell target cells, other healthy skin cells, that it's time to go ahead and start dividing. So you can see that the growth factor will bind to what we call a receptor protein, and then there will be a series of chemical reactions called signal transduction pathway, which will lead to a gene being turned on. And we'll learn more about genes being turned on and off later this year. But basically, like we learned with lactose intolerance, there can be switches to turn genes on and off. And so the binding of this growth factor will eventually lead to a switch that turns on genes that are involved with the cells doing the next activity. So if it's in G0, for example, it might then tell the cell to go ahead and do some more G1 activities, or to start making the proteins needed for S, which is DNA replication. So we can see the genes get turned on, proteins are going to get made, and then the cell is going to be basically doing the cell cycle now so that it can make new cells to replace the injured, damaged cells in your cut. Now, another type of external signal is going to be more long-term. Okay, it's not necessarily going to be a short-term response, like an emergency with a cut. Instead, hormones are produced in a gland, and then they travel to a target cell through the blood. And because they're kind of more of a slower progress than, say, growth factors, which are released between neighboring cells, since hormones have to potentially travel far through the body, it leads to kind of a slower, more sustained response. And so we see hormones being involved with growth and development, like growth hormones, or even sex hormones like estrogen and testosterone are involved in growth and development. So let's talk a little bit about an example with hormones. Okay, so a hormone is going to be made by a producer cell, and that's going to be the gland. So you have glands throughout your body. You have glands in your brain, like your pituitary gland. You also have glands in your reproductive organs, like your ovaries and testes, which make hormones, okay? And they're going to release the signals, which in this case are hormones, okay? And they're going to travel through the blood to whatever target cell it might be, and then it's going to trigger those target cells to turn on genes that will lead to cell division. Okay, so let's look a little bit about our reproductive hormones. And I'm going to use female hormones as an example. So let's say the gland that we're going to look at is an ovary. The ovary is going to produce the hormone, in this case, which is estrogen. And estrogen can then bind to target cells in breast tissue. And it can tell then the breast tissue it's time to divide and grow. Now we will see later on in the next unit that this pathway with hormones can sometimes go wrong. And the cells can sometimes make too much estrogen, or perhaps the breast cells have too many of these receptors, and so they're extra sensitive. And as a result, this breast tissue might grow too fast, or it might grow out of control, in which case you might get breast cancer. So we'll talk more about that when we get to the cancer section. But understand that growth factors and hormones communicate with cells and tell them it is time to divide. There's a need for the body to grow or repair. All right, our next one was then, how do cells know that they're progressing through the cell cycle correctly? They have to go through G1, S, and G2 and M. How do they know they're doing everything right? Well, we just talked about the fact that they're going to go through G1, they're going to grow, and then they might hang out in G0, okay, until they're called to do so by a growth hormone or growth factor. So then it needs to then progress on to S. But before it can progress on to S, there's a checkpoint. And checks and make sure that the cell's okay. There's also a G2 checkpoint to make sure that everything happened well in S and G2. And there's an M checkpoint to make sure that mitosis is happening correctly. So let's go through each of these checkpoints and talk about them. So there are 3 checkpoints of the cell cycle. So in this case, we're going clockwise. Okay. So G1 checkpoint checks for a certain amount of stuff to make sure that everything is going correctly. So G1 checkpoint checks that there's the correct number of nutrients, so I like to think of that as it's checking for growth. Okay, it's also going to check, like we just talked about, for growth factors. There's a message, it might also be hormones, that there's some sort of signal that it is time to divide. And then another thing it's going to check for is DNA damage. So let's go back to the example of your skin, right? So your skin is on the surface and it's constantly bombarded with all kinds of environmental factors. So it might be experiencing damage from like UV light or drying out in the air. And so your skin cells take a beating, okay? And the UV light that you're exposed to can damage the DNA. And your body doesn't want to go ahead and copy damaged DNA in the S phase. So it's got to make sure that everything is okay. So what it's going to do is it's going to check that the cell's the right size. It's going to check and make sure that the DNA is okay. And then if everything looks good, it can go through the checkpoint and go to the S phase. If things aren't good, if it hasn't grown enough, it'll go to G0 and grow some more. If the DNA is damaged, the cell will either try to fix it or it will commit apoptosis, so cell suicide. Because if there's too much DNA damage, it doesn't even want to try to fix it all. It'll just say it's better just to replace it with a healthy cell rather than trying to replicate a damaged cell. All right, so let's say it does everything correctly there. Next up, DNA synthesis. Okay, that's going to be DNA replication. Okay, and then it's going to progress through and it's going to hit G2 where it grows. And again, G2 checkpoint is going to check for cell size, Got to make sure there's enough organelles, proteins, enough cytoplasm to make 2 new cells out of 1. And it also checks DNA replication. So it wants to make sure that the DNA got copied correctly in the S phase before it gets divided up. And again, like I mentioned it before, it'll either fix mistakes or it will have the cell commit suicide if there are just too many mistakes. So let's say the cell is the right size, it copied it correctly, then it's going to go into the M phase. So remember, M phase, mitosis, prophase, metaphase, anaphase, telophase. Metaphase is when the next checkpoint is, and it wants to make sure that the chromosomes are attached to the spindle. So if you remember, chromosomes have condensed at this point, and they are connected to the spindle, and the spindle is going to help pull apart those sister chromatids so that one goes into each of the daughter cells. Now, if the spindle isn't done correctly, so let's go over here to this side and draw it out. Let's say the spindle doesn't attach properly. Well, the chromatids might not separate properly. And basically all the DNA might go into one cell, and this cell over here is missing DNA, and that would be bad. So the spindle is really important for ensuring that the chromosomes get divided equally. So if that's correct, then it can go ahead and move through. So I mentioned this before. If the cell can't meet the requirements of a checkpoint, what exactly does it do? Well, it's going to try to fix it first. It's going to try to repair it, or it'll die. Okay? So the repair might be something like growing more or attaching the spindle correctly. But if it's DNA damage or DNA replication errors, then the DNA has to be fixed. Okay, so it says if there's DNA damage or DNA didn't replicate properly, the cell tries to repair the DNA molecules so it can pass through the checkpoints. So here you can see DNA repair. So here we can see there's a mutation. A G got paired with a T. So maybe in DNA replication there was a mismatch is what we call it. So what's going to happen is the cell will basically cut out a chunk. It'll cut out the mismatched nucleotide and some more around it. And then DNA polymerase 2, DNA polymerase 2, okay, it's going to go in and it's going to fill in that gap. So we learned about other DNA polymerases, right? We learned about DNA polymerase 3 being our main builder, DNA polymerase 1 being our primer fixer. Well, DNA polymerase 2's job is to fix mutated DNA. And then once it's done, okay, we have the correct match and everything looks good. So the cell's going to do this, and its cell knows that there's a mismatch because G and T don't bond, right? And they're not going to form the correct hydrogen bonds. So instead of having this nice equal diameter, there's almost like a bump. You can see it here. It's like straight, straight, straight. Everything looks good, and then it's like bump. And so the cell can find these bumps in the DNA molecule where something isn't paired correctly, and it will cut out those bumps, those mismatches. So repair or die, right? It's going to check itself before it wrecks itself. It's got to fix things. Okay, so repair, we just learned about, die. So if the DNA damage is too extensive, the cell's going to kill itself through a process called apoptosis. So apoptosis is sometimes called cell suicide. It's sometimes called programmed cell death. So this is going to occur basically if there's DNA damage or if something called the telomeres at the end of chromosomes are too short. More about that in the next video. So in apoptosis, okay, if there's DNA damage or if there's going to be DNA damage, what the cell will do is it starts a series of chemical reactions that lead to the digestion of the cell. So the cell loses cell size, the DNA starts to condense, okay? The membrane starts to break apart. They call this blebbing, okay? So the lipids start to break apart, and then you can see that it's really like blebbing. It's breaking up into these little broken pieces, these little broken cell pieces. And what comes along is this white blood cell called a macrophage comes and gobbles up those blebs, those apoptotic bodies, these little pieces of cells, and gobbles them up so that it's digested and it's out of the way. So basically the cell cleans up and gets rid of any mistakes it might have. So we learned about the checkpoints. We learned about some of the consequences if checkpoints aren't made. But how does a cell exactly know that it's meeting the checkpoint requirements? How does it know if it can actually go through a checkpoint? Well, the answer there are some chemicals. These are internal signals, and they are cyclin and cyclin-dependent kinases, also called CDKs, and they control the cell cycle. Okay, so cyclin-dependent kinases are a series of enzymes that add phosphate groups to proteins. Okay, and CDKs, or cyclin-dependent kinases, are inactive. Okay, so they're basically in the off form. Only when cyclin binds, which you can see here, does it turn on. So this is off. When cyclin binds to CDK, the enzyme changes shape and it turns on. And now cyclins and CDKs can turn on target proteins. So this is a target protein and it can go do an important job for the cell. When that is done, cyclin breaks down, but CDK, because it's an enzyme, can get used over and over again. So basically, that's the main steps. Well, how does that fit into the checkpoints? Well, cyclins are these proteins that get made as the cell does proper cellular functions. So as the cell does normal G1 activities, cyclins get made. Okay? As DNA replication happens correctly, cyclins get made. So these cyclins are building up throughout the cell cycle. And when there's enough cyclins, they will bind to CDK, they'll turn them on, and then they'll make a whole bunch of activated proteins that can go off and do jobs. So let's say this cyclin was a G1 cyclin. It's going to bind to CDK, and then it's going to activate target proteins that are going to help with the S phase. So in this case, this target protein is now active. It can go do a job. Maybe it's involved in DNA replication. So basically, think of cyclins and CDKs almost as like these switches that turn on the enzymes needed for the next step. So here you can see an additional example. Notice the CDKs stay constant throughout this cycle, but there are different cyclins that are being made. So here, for example, as G1 happens correctly, G1 cyclin is made. It will then bind to CDK, and it will make start enzymes that help with the S phase. And then we break down that cyclin because we're done with it in our bodies. And then as S phase happens correctly, S phase cyclin builds up, it binds to the S phase CDK, and it's going to then activate some proteins that maybe help it move on to G2. Okay, and so we see kind of these progress of these different cyclins occurring. So there are these different cyclins that happen throughout the cell cycle, as you can see in this graph, and they kind of rise and fall with each checkpoint. So there's a checkpoint in G1, there's a checkpoint in G2, and there's a checkpoint in M. So having the right amount of cyclin at the right time tells the cell, hey, everything looks good. We've built enough cyclin, therefore every activity must have occurred. We replicated the DNA properly, the DNA doesn't have damage, the cell's grown correctly, or maybe in the M phase, the spindle's attached, okay? And everything looks good and we can go through these checkpoints. And with that, that is how the cell knows how to do it correctly. We will be examining cancer and we'll understand that these checkpoints and apoptosis and all these regulatory mechanisms get messed up and mutated when cancer develops.